Good day and welcome. On today's biology class, we are going to look at a very interesting topic known as the cell and its environment. Okay, this topic is critical in the JAMS UTME and also in the Senior School Certificate exam conducted by the West African Examination Council. We also have other exams that students sit the biology subject and they are asked questions in this section. Let's go. Now consider the question on the board, UTME 1978, question 41. It says a Spirogyra cell was placed in a solution X, and after 1 minute, the cell had been plasmolysed. And then they want us to tell what solution X was. Okay, so we'll get back to this question. I just needed you to see that it is tested often in exam situation. Okay, so we have to understand certain concepts like what is an environment, what are the types of environments, what are the effects of these environments on both plant and the animal cells respectively, and what is the best environment for a cell. Okay, so let me start by saying that a cell is the structural and the functional unit of life. That is to say, cells are the basic units of existence. All right, just like your atoms are the basic units of matter. So your cell is likened unto the basic units of existence. Okay, now what is an environment? Now we all know what an environment is. An environment is a surrounding. Okay, so when we say a cell and its environment, we are talking about the interactions that occur between a cell and its surrounding. Yes, okay. So usually the surroundings of cells, okay, are often in liquid form. Okay, they are often in liquid form. All right, we have what we call cytoplasmic content. Yes, so most cells exist in watery environments. Let me use that language. All right. Now, in mature animals, okay, or advanced animals, our cells are surrounded by blood. That is to say, they are bathed in blood. All right. So that is the environment of our cell. Excellent. Now, we know what an environment is. We go to point 2. What are the types of environments? Okay, so in relationship with the content of a cell, we have 3 types of environment. 1, we have what is called a highly or very concentrated environment, which is termed hypertonic. Okay, highly concentrated environment is said to be hypertonic. Okay, now we have an environment that is less concentrated compared to the content of the cell. Okay, so when an environment is less concentrated, such an environment is said to be hypotonic. I hope we're learning. Excellent. Now, we have environments that are in a state of equilibrium. That is to say, the environment and that of the cell content itself, okay, none is higher or less than the other. Yes. So whenever the concentration of an environment and that of the cell is in equilibrium, we describe such an environment as isotonic. Okay. So let's call it stable concentration. We describe that as isotonic. So let's take that again. If the environment is highly concentrated when compared to the cell content, it is said to be hypertonic. If it is less concentrated than the cell content itself, it is said to be hypotonic. If it is in equilibrium, that is to say, it is not higher or less than the content of the cell, rather they are equal in concentration, it is said to be isotonic. All right, so we are done with outline 2. Outline 3, we are going to see the effects of these environments on plant and animal cells. Now, before we proceed, we need to understand something very basic, but most people fumble on this. Now, what is that? The plant cell and the animal cell are characteristically different, okay? And speaking of this characteristic difference, we focus on the presence of the cell wall in plants, okay, and the absence of a cell wall in the animal cell. All right. Now, you know the plant cell has a cell wall, the animal cell has a cell membrane. Plant cells equally have cell membrane in addition to their cell wall, but animal cells do not possess a cell wall. Okay, so based on this, their interactions with their environment is going to differ a great deal. Yes. Now, the cell wall of plants, for example, is made up of cellulose, which is a very flexible material and gives the plant cell rigidity. All right, it gives the plant cell rigidity. Okay, now this cell wall is absent in the animal cell. So, like I said, their interactions with their environment is going to differ. Okay, so let's have a good look at that. Now, when a plant cell, let's start with the plant cell. When a plant cell is placed in hypertonic solution, that is a solution that is highly concentrated, a solution whose concentration is more than that of the cell. Now, when a plant cell, we're going to see how each of these two cells react in these environments. When a plant cell is placed in hypertonic solution, all right, what happens is, remember, hypertonic solution means the concentration of its environment is higher than that of the cell. Okay, in hypertonic, the concentration of the environment is higher than that of the cell. So what happens here when a cell is placed in such an environment, all right, is that the cell starts to lose water. It begins to lose its content to the environment. Okay, so when this starts to happen, remember in the plant cell, what you have mainly in the plant cell are large vacuoles whose concentration, okay, we have ions, we have water, and we have other things present in the cytoplasm of cells, present in their vacuoles. Okay, so when the plant cell is introduced to a hypertonic solution, a solution whose concentration is higher compared to that of the cell starts to lose water to its environment, to the surrounding, okay, through a process we call exosmosis. Exosmosis. Okay, this is how the cell loses water. All right, so as it loses water, what happens then is that there starts to occur what we call shrinkage. Yes, the cytoplasm, the content of the cell itself starts to shrink. All right, it starts to shrink. Good. So that shrinkage is what we term plasmolysis. That shrinkage is what is called plasmolysis. Now, this is what happens when a cell is placed, a plant cell, mind you, is placed in a highly concentrated environment. All right. Now, when a plant cell is placed in a less concentrated, that is what we call hypotonic environment or hypotonic solution, what happens then is that the cell starts to take in water from its environment. It starts to take in water from its environment. All right. So this process of taking in water is what we call endosmosis. Endosmosis. Okay. So don't forget, when the cell loses water, it does so by a process called exosmosis. And when the cell gains water, it does so by a process called endosmosis. All right. So when the cell starts to take in water, what happens here, please stay with me, is that in plant cells, the cell starts to swell because it is taking in water, so it starts to increase. Same thing happens in animal cells, but the end product or the aftermath of this swelling is different. In plant cells, when they are placed in hypotonic solution and then they take in water by endosmosis and they start to swell, all right, the cell does not burst. You know, you could swell up to a point and then you burst. The cell does not burst. This is because of the presence of what we call the cellular cell wall. So in plant cell, because of the cellular cell wall present, no matter how much water is taken in, the cell only swells but never explodes or never bursts. Yes. Okay, so this is because of the presence of the cellular cell wall. All right. Now, when this happens, however, the cell enters into a stage we call turgidity. Yes. So when plant cells are placed in hypotonic solution, what happens to them is that they become turgid. So because they are swollen, all right, a form of pressure known as the turgor pressure, okay, becomes accumulated or is present. We call it the turgor pressure, turgor. All right. But the cell, the plant cell does not burst. Yes. So this phenomenon is what we call turgidity. It is known as turgidity. All right. Excellent. So we have seen what happens when a plant cell is placed in hypertonic. It suffers what we call shrinkage. We have seen what happens when a plant cell is placed in hypotonic. It suffers what we call swelling. So when in hypertonic solution and the cell shrinks, that is termed plasmolysis. When in hypotonic solution and the plant cell swells but does not burst, that phenomenon is called turgidity. Excellent. Now, for the animal cell, remember they do not possess cell wall, rather they have a cell membrane. For the animal cell, when the animal cell is placed in a hypertonic solution, all right, the animal cell loses water by exosmosis. As it loses water, it shrinks. Yes, it shrinks. All right, that is to say, it ruptures, it shrinks. Yes, all right, and it results to a phenomenon which we call crenation. So when a plant cell loses, not plant, sorry, an animal cell loses water or its content to a hypertonic environment, what happens to the animal cell is what we call crenation, shrinkage. Yes, the shrinkage of the animal cell when placed in hypertonic solution is known as crenation. All right. Now, when an animal cell is placed in hypotonic solution, hypotonic, all right, what happens to the animal cell is that it takes in water, it gets swollen, but because it has no cell wall, it will eventually burst. Yes, it will get ruptured, it will burst. All right. Now, this is what happens in what we call the red blood cells. Yes, which is present in animals, the red blood cells. Now, you know that a mature red blood cell has no cell wall. It does not have a cell wall. A mature red blood cell, yes, has no cell wall. All right. Now, when the red blood cell takes in water and gets swollen, it will eventually burst. This phenomenon, okay, in red blood cells is what we call hemolysis. Hemolysis. All right. So be careful of these terms we are using: exosmosis, endosmosis, plasmolysis, turgidity, crenation, hemolysis. All right. So they are basic terminologies. Exosmosis is the process by which water leaves the cell. Endosmosis is the process by which water enters the cell. All right. Now, plasmolysis is what happens to plant cells when they are placed in what we call hypertonic solution. Hypertonic solution. All right. Then, when a plant cell is placed in hypotonic solution, it gets turgid. Yes, resulting in what we call turgidity. All right, and don't forget the pressure that is now obtainable within that swollen plant cell is what we call the turgor pressure. Turgor pressure. All right. Now, I also mentioned that for animal cells, when they lose water, that phenomenon is called crenation. So when placed in hypertonic solution and they lose their water, their cell content to their environment, all right, such a phenomenon is called crenation. And I said when placed in hypotonic solution, the animal cell absorbs water and then swells. But because it has no cell wall, it eventually ruptures. Yes. And I said this happens in red blood cells, and that phenomenon is termed hemolysis. Excellent, excellent. So you get that. Now, we have a final outline here which says what is the best environment for a cell? The best environment for a cell is a stable environment which we call isotonic. So whether a plant or animal cell, whichever it is, if it is placed in isotonic solution, that is a solution that is not overly concentrated or less concentrated than itself, being the cell, okay, such a cell is going to function properly. Yes, it's going to function properly. So the best environment for cellular activity, cellular operation, or for the cell, generally speaking, is the isotonic environment or the isotonic solution. All right. Now, I'm going to mention here that part of how things, water, and other components move between the cell and its surrounding, okay, involves what we call diffusion, osmosis, all right, and what we know as active, passive, and bulk transport. Yes, okay, this is what some biologists term cell transport. All right, now in humans, we have a circulatory system. We have arteries, veins, capillaries, arterioles, and so on that help to carry both blood, water, ions, and other materials around the body. Okay, in plants, we also have their cell network, yes, or transport system, which involves what we call vascular bundles. Okay, with the two main bundles being the xylem and the phloem. There are others. Okay, now in the cell, for transport of materials to take place, I am saying to you that we have three types of transport: active transport, passive transport, and what we know as bulk transport. Okay, now we also have the operations of diffusion and osmosis within the cell. Yes, okay, so actively speaking, we have diffusion and we have osmosis. Now, what is diffusion? Diffusion is simply the movement okay, of gaseous particles, all right, from a region of higher concentration to a region of lower concentration, with or without the presence of a semi-permeable membrane, all right, that is a selectively permeable membrane. Osmosis, on the other hand, is the movement of water molecules from regions of higher concentration to a region of lower concentration, usually via a semi-permeable membrane. All right. So don't forget that there are two types of osmosis, the endosmosis and the exosmosis. Endosmosis is the taking in of water, the movement of water molecules from the environment of the cell into the cell. Exoosmosis is the movement of water molecules from within the cell itself to its environment. Okay, now let's come to this question. So a spirogyra cell was placed in a solution X, and after 1 minute, the cell had been plasmolized. Observe the terminology is being used. Your spirogyra is not an animal, it's a plant. Excellent. So what they are telling you here is that a plant cell was placed in a solution X, and after 1 minute, plasmolysis occurred. Now, when does plasmolysis take place in plant cells? Do you recall? When does plasmolysis take place in plant cells? Plasmolysis takes place in plant cells when they are placed in hypertonic solution. That is a solution whose concentration is higher than that of the cell. So the cell will lose water, will lose contents to the environment, thereby shrinking. Okay? So it means that solution X has to be highly concentrated. Do you get that now? For the plant cell to be plasmolized, it means solution X was hypertonic. So option A says tap water. Option B says a very strong sugar solution. Option C says a weak sugar solution. Option E says distilled water. Please take note, distilled water is a hypotonic solution. It is not hyper. Distilled water is hypotonic. Yes. And when a cell is placed in hypotonic solution, the cell takes in water, swells up, but does not burst because it has a rigid cell wall. All right. So what happens when a cell is a plant cell, mind you, is placed in distilled water is that the cell becomes turgid. All right. And then a form of pressure known as the turgor pressure is found within that cell. Yes, because it is swollen. Okay. Now we have a very weak salt solution. So what do you notice? Weak means less concentrated. Weak here again, less concentrated. So option C and D, they are off. I already told you that the distilled water is hypotonic, meaning less concentrated than the cellular environment. Okay, so it goes. Now, our best bet will be option B, which is a very strong sugar solution. This expression can be summed up in one word, which is a hypertonic solution. I believe you get that. Excellent. So don't forget what an environment is. It is the surrounding of a cell. Don't forget that there are three types of environments. A highly concentrated environment, which is hypertonic, a less concentrated environment, which is hypotonic, and a stable environment, that is an environment which its content or concentration and that of the cell are equal, which is isotonic. Okay, remember that a plant cell and an animal cell will react differently in these environments. When a plant cell is placed in hypertonic solution, the plant cell loses water to its environment, okay, thereby shrinking. That phenomenon is called plasmolysis. When an animal cell is placed in hypertonic solution, okay, it loses water and then it shrinks. That phenomenon in animal cells is called crenation. Okay. Now, when a plant cell is placed in hypotonic solution, that is a solution whose concentration is less than that of the cell, what happens to the cell is that it takes in water it gets swollen but does not burst. It does not rupture. Okay? Rather, it builds what we call turgor pressure. Yes. And then the cell is now in a state called turgidity. All right? But when an animal cell is placed in a hypotonic solution, all right, and it takes in water and gets swollen, because it lacks a cell wall, it will eventually burst. Okay? And I gave a typical example using the red blood cells. Yes. How that when a mature red blood cell is placed in hypotonic solution, such as distilled water, it will swell to a point that it will burst. And when it does so, this phenomenon is what I termed hemolysis. All right? Excellent. Now, we have a certain phenomenon known as flaccidity in biology. Flaccidity. Now, it occurs mainly in plant cells. All right. So, this phenomenon is used to explain a condition that occurs during drought. That is when there's dryness or absence of water for plants in the soil. Yes. So, when a plant cell loses water more than it absorbs, when a plant cell or a plant loses water to its environment more than it can absorb from the soil, flaccidity takes place. That's what we mean by flaccidity in plant cells. All right. So I'll stop here. I will advise that you get a past question book on the exam, whatever exam you're preparing for, on the subject biology, and attempt questions on cell and its environment. Do ensure you watch this video over and over again so that you can get the message. It's basic and it's simple. Until next time, this is Covenant Science Tutorial Channel.